Hello, everyone. Thank you all for joining to listen to my poster talk titled Investigation of Metallo Drugs as Antimicrobials. To begin with, I would like to introduce you all to PDI, a light trigger therapy that can act as effective alternate strategy to kill the pathogens, including antimicrobial resistant species. If you look at this figure one on top, it can help explain PDI in a detailed way. A photosynthetizer is a molecule which is designed to be light activated by absorbing energy in its ground state. And upon absorbing this energy, it can be converted into an activated and excited state. Now, this molecule wants to go back to its ground state, which it can do by either of the two pathways. One is by transferring that extra energy to the through the type 1 reaction, which is an electron transfer process, in which it can react with the environment, in which it can react with the molecules present in the environment by converting them into reactive molecular species such as hydrogen peroxide, superoxide, anion, and hydroxyl radical. Another pathway is by energy transfer reaction, in which it can react with the molecular oxygen present in the environment and convert it into a cytotoxic form, which is a singlet oxygen species. Now, these singlet oxygen species and the reactive molecular species, they're cytotoxic in nature and they can kill the pathogens, including the antimicrobial resistant strain, because they produce a rather non-specific immediate burst of these species, which can that then can be controlled spatio-temporally to regulate the treatment regimen that we want to design. In today's poster, I'm talking about ruthenium-based coordination complexes to which two functional ligands are attached, and these two functional ligands has a fluorine-containing methyl groups attached to the two bispyridine co-ligands. A third ligand is known as the functional ligand, which is attached to increase the lifetime of the molecule by extending by doing its pi expansion. As you look at the pharmacokinetics of these molecules, which is determined by lipophilicity determination, which refers to the compound, whether the compound is preferring the lipophilic phase or the aqueous phase, it, it can be seen that the increase in the number of thiophene chain length from n is equals to 0 to n is equals to 4 here, it's leading to increase in the lipophilicity of the molecule. Next, we also studied the photophysical characterization of these complexes in order to calculate their energies and the excited state kinetics and the lifetime of various excited states, namely MLCT, metal to ligand charge transfer state, and a triplet ILCT, an intraligand charge transfer state. We also looked at the singlet oxygen quantum means of these complexes because they are important cytotoxic species on which the PDI might rely on, and we might need that information to further analyze what's happening with our complexes. We also studied the photobiological activity of these complexes by comparing the photoantibacterial activity we obtained for Enterococcus faecalis, a gram-positive bacterial strain, uh, a gram-positive bacterial species for which there are two strains available. One is 29212, which is an antibiotic susceptible strain for which the plots are on the left side. Another is a V587, a vancomycin resistant antimicrobial resistant strain obtained from a UTI patient for which the plots are shown on the right. As you can look at the plots, you can see that the activity for the complexes we designed, it's all the complexes in the family are not showing activity, but the main point is that most of the complexes in this family are showing us activity in both antibiotic susceptible and antimicrobial resistant strain. The top plots are showing us the EC90 values, which is the concentration at which 90% cell death is happening, whereas the bottom plots are showing us phototherapeutic index value because we want to compare the activity under light and dark conditions. We also are looking further to analyze the localization and the cell uptake studies for the development of the structure activity relationships based on various parameters such as physicochemical, photophysical, and photochemical. Last but not the least, I would like to acknowledge the Department of Chemistry and Biochemistry and UTA, and also the NSF award that we received to fund this research. Thank you so much for listening to this talk.